Well, hello everybody and welcome this morning for our virtual webinar. Today we're really excited to host Grace Jia. She is a research fellow in the lab of Dr. Andrew Gruver. And we're really excited because this is our first a webinar on plant sectioning. So today we're going to feature Grace Jia. She is an ORISE research fellow in the lab of Andrew Gruver at the USDA Forest Service Northern Research Station. And Grace brings a host of expertise. She graduated from the University of Vermont with a Bachelor's of Sciences in Wildlife Biology, and then she got a Master's in Biology from Tufts University. The Gruver Lab focuses on utilizing genetic and genomic tools to unravel the growth and physiology of trees. And their projects include investigating genetic regulation of wood formation in response to drought, stress, developing proteomic approaches for studying wood traits, and combating invasive tree pathogens. So in this webinar, Grace here will dive into the Gruver Lab's work on the American chestnut trees and the analysis of woody plant tissues. And before we begin, we have some more people to let in from our waiting room. And I just want to say that if any of you have any questions or comments, please feel free to utilize the chat box on Zoom. And I'll open up a roundtable for Q&A at the end of Grace's talk. With that, I want to say, Grace, I'm going to hand over the microphone to you. And thank you so much for being here. And also a shout out to Dr. Gruver, who is also here as well.
Yes, thank you so much, Abby. Good morning, everyone, and thank you for joining us this morning. As Abby has already stated, my name is Grace Jia, and today I will be speaking about how my lab, Dr. Andrew Gruver's lab, who is also in attendance, as Abby just said, is using the Compresstome as a tool to investigate American and Chinese chestnut histology. So I know that Abby has already done a really great job of introducing me already, but before we get started, I wanted to just kind of summarize my experience and share that with you all. So I'm currently a research fellow through the Oak Ridge Institute of Science and Education, and my position is with the USDA Forest Service Northern Research Station in the lab of Dr. Andrew Gruver, based in Burlington, Vermont. I obtained my undergraduate degree from the University of Vermont in wildlife biology, and then obtained my master's at Tufts University in Medford. I'm also happy to share that I'll be starting my doctoral studies back at the University of Vermont this fall in the Department of Plant Biology. My research interests are at the intersection of microbiology, ecology, and genomics, and I'm very interested in using tools from molecular biology and ecology to investigate plant biodiversity that will then be used, hopefully, in conservation management and practices. And with that, here's an outline of my talk today, starting with a brief summary, starting with a brief history of American chestnuts, a summary on chestnut blight and pathogenesis, the project scope, a quick lesson on chestnut tree identification methods, and the histology study that I'm working on in the Gruver Lab. And on the right side of this slide, you can see an image of the American chestnut trees that our collaborators, the American Chestnut Foundation, are growing in the greenhouse at our lab in Burlington. To begin, I would like to talk about the brief history of chestnut trees and its introduction into the eastern United States. American chestnut trees have a strong ethnobotanical connection to the eastern United States. Prior to the 1800s, there were around 4 billion trees spanning this part of the country. Original inhabitants of the Appalachians coexisted with American chestnut trees by providing a rich environment for them to grow while also utilizing their nuts as a food resource. Chestnut trees were not only important to humans, but also to other flora and fauna in the environment, as they provided dense calories, vitamin C, antioxidants, and essential plant nutrients from their nuts and leaves. With the arrival of European settlers and the displacement of indigenous peoples, it was then discovered that these trees were a suitable resource for lumber and building materials, as the wood is rot resistant and straight grained. The nuts were also continued to be used as a food source and was sold near and far. Unfortunately, due to the introduction of chestnut blight, also known as Cryphonectria parasitica, also commonly known as chestnut canker, the abundant chestnut dominated forests we once knew began to change rapidly. Chestnut blight, to give you a little bit of a background, is an ascomycete, and ascomycetes are currently the largest phylum of fungi with over 64,000 species observed today. This type of fungus, also known as sac fungi or spore shooters, can produce sexually and asexually. It was first introduced to the eastern United States from East Asia through infected chestnut plants. And to provide a little bit of an overview of how this pathogenesis occurs, it follows a few steps. So first, spread. Spores of infected trees are released into the environment and are spread via wind, water, or transferred through direct contact from orange stomata. And this orange stomata you can see on the left side of the slide. Next, infection. Spores infect chestnut trees by entering through wounds in the bark. Then the growth of the fungus occurs by the fungus entering and growing within the inner bark and cambium. And at this stage, it produces mycelial fans. It then develops and colonizes within the tree, and cankers develop as the fungus continues to grow. And an example of that is the image on the right. You can see the external part of the tree growing the canker. The final step is girdling and killing the stem or branch that it has been infected. So the fungus will girdle and kill the cambium and continue to do so after a stem or branch is killed. So just to show you how intense this chestnut blight can alter the American chestnut tree. Here is a picture just to show how large and dominant American chestnut trees once were. And this image shows Jim and Caroline Walker Shelton's family standing underneath an American chestnut tree below Tremont Falls in the Great Smoky Mountains National Park along the border of Tennessee and North Carolina. And this image was taken circa 1920. So a lot of the chestnut trees that we see today are not nearly to the scale or size as what we see on the left side of this slide. This short video shows the expanse of pathogenesis over time, depicting the introduction of the pathogen and how rapidly it developed in the eastern United States. So as you can see, its range is completely covered today. And again, here is just another picture on the side showing the orange stomata that is part of the developing canker. So the project scope and what we are trying to find out in the Groover lab is to investigate and compare if stem anatomy, cell morphology, or biochemical differences between disease-susceptible American chestnut and disease-resistant Chinese chestnut trees may influence chestnut blight susceptibility. The plan is to carry out this project using histology to understand the basis of susceptibility versus resistance between these species, and we will do so by inoculating both American and Chinese chestnut seedlings with the chestnut fungus, which is Cryphonectria parasitica, and observe the infection using a time series. From this, we will then compare gene expression using RNA-seq. And just to reiterate the histology component of this, we will also be using a combination of different fluorescent stained dyes to understand changes in cell walls and biochemistry. Again, this is a project in collaboration with the American Chestnut Foundation, and we are very excited to be working with them on this project. So before we get started on utilizing the Compresstome for our histology research, I wanted to dive a little bit deeper into the morphological differences of these trees, specifically looking at their leaves, buds, stems, and stipules. So as you can see on the left, we have American chestnut, and on the right, we have Chinese chestnut. And the American chestnut leaves are long in relation to its width. They have large prominent teeth on the edges and bristles at the end of each tooth that curve inward. The base of the leaf blade tapers sharply, and the leaf is very thin and paper-like. If you are able to get your hands on a chestnut leaf, that will become a lot more evident if you can hold it and feel it. However, the Chinese chestnut leaf is oval-shaped, the teeth are much smaller, the base of the leaf blade is rounded, and these leaves are a lot more thick and have a waxy feeling. When looking at the buds and stems, the buds of the American chestnut are pointed and they angle away from the stem, and the stems are smooth and hairless. And the color of the American chestnut stems are reddish brown to dark green, as shown in the image on the left. And they are small but numerous lenticels on the stem. When compared to Chinese chestnut on the right, they have more rounded buds that hug the stem. They have hairy stems and hairy leaf veins. The stem color is a little bit more tan to a pea green, and they have large lenticels, which are the bumps on the stem. And finally, looking at the stipules on an American chestnut are slender and angle sharply out from the stem, and they usually fall off in June, whereas the stipules on a Chinese chestnut are broad, they're covered by the buds, and they actually remain on the stem through to September. So those are just some basic external morphological differences that you can observe from your eye. Because those are only external morphological differences, we are using the Compresstome vibrating microtome in order to investigate histology. And of course, this tool is made by Precisionary Instruments, who have so graciously invited me today to talk about the research in the Groover Lab. While there is some published research on the use of the Compresstome vibrating microtome for plant histology, as far as we know, no one has used this tool to investigate woody stems or trees before. In order to adapt the use of this machine for our histology project, it is important to investigate the following. How does the Compresstome perform when cutting wood stems? What are the best settings to use? And what factors need to be considered in order to obtain suitable samples? We tested these questions and started developing our methods. So to begin, I would like to talk about the methods that we had tested out and thought about when obtaining these cross sections. So when sectioning woody tissue, the following factors are important to consider. Speed and oscillation of the Compresstome, which a recommendation from Precisionary Instruments can be seen at the bottom of this slide. Firmness of agarose for embedding, whether it is necessary to embed woody stems in agarose or if you can use the Compresstome without embedding a sample. The length of the woody stem sample and the thickness of the cross-section that you would like to obtain for microscopy. Since we are using plant stems for this investigation, we use these parameters provided by Precisionary Instruments as a basis for developing our methods right here. So as you can see, speed and oscillation, 3 to 4, 4 to 5, and the suggested percentage of agarose being 2%. Through project experimentation, we discovered the following. The basis of speed and oscillation that was recommended by Precisionary Instruments ended up working very well for the type of woody tissue that we were sectioning. In terms of embedding in agarose, tested 0%, which is no agarose, 2%, 2.5%, and 3% agarose firmness. The suitable firmness for sectioning stem tissue, if you wish to embed it in agarose, is 2.5%, which is just a little bit over the recommendation that Precisionary Instruments had discovered. But also samples not embedded in agarose produce likewise results. Because the length of a sample, or because the length of a sample, because stems can be quite thin, the length of the sample is also a very important factor, and it was discovered that about 1 centimeter or shorter as a width is suitable length, as the width of the tissue is too thin despite being embedded in agarose and will deflect from the cutting blade. The thickness of the cross sections was also investigated and tested between 50 micrometers and 70 micrometers, and found that the suitable thickness for a cross section in order to mount it and prepare on a slide for microscopy was 50 micrometers. So this is an example of a American chestnut cross-section that I obtained using these parameters. And just to give you a little bit of a lesson on anatomy, these American chestnuts are ring porous, which means that the vessels / pores vary in size depending on their developmental stage. The pore sizes are distinctly larger in early wood, which you can see in this image, where they are smaller and more compact in later wood development. This cross-section was sampled from an American chestnut tree grown by our collaborators at the American Chestnut Foundation that was planted in 2022. So still quite young in its development. In this picture, the anatomical components are labeled showing the epidermis, cortex, fibers, secondary phloem, vascular cambium, secondary xylem, and the pith. We have to remember that ultimately the fungus will be entering the stem and killing the cambium later, and factors such as the thickness of the tissue, the composition of the cells within each of the tissues, certain wall types, and other biochemical differences will hopefully aid in explaining the differences in susceptibility of the infection in the American chestnut and the resistance in Chinese chestnut. So here is that same image from the previous slide showing a 20x magnification of the three, as well as 3 components from the 10x image. These images show a clear preview of what anatomical differences we may be looking for. This is an example of almost the same image, but instead of the American chestnut, it is the Chinese chestnut, showing a 20 times magnification of 3 components from the 10x image, except, like I said, in Chinese chestnut. As you can see from these images, as you go up in magnification, the thickness of the sample limits the ability to get images with good resolution. That is very evident in this part over here, where it's a little bit more blurry, and over here, where it's a little sharper. You can see at the bottom of the 10x magnification that fully intact cross-sections are needed in order to carry out this investigation. There is a tear along the cambium layer, and these cells are very delicate and have cell walls that are still dividing as the wood is lignified. That's just something to be aware of when you are taking such cross-sections from these samples. And here's a side-by-side comparison of the American and Chinese chestnut images that I have taken in our lab on a preliminary level. And again, this is just a preliminary comparison. A more accurate observation will be observed using our histological stains, but you can see that there is already a difference in color. The cortex looks quite different, with the American chestnut showing larger diameter cells that look more prominent, and the epidermis and layer of phloem fibers on the Chinese chestnut look a little bit thinner. Because of these preliminary comparisons, we are still developing our methods for our histological study. And we are doing that by adapting the protocol, the staining protocol from this paper that was published by Ursachi et al. In 2017. This paper investigates the use of ClearC, which is a solution, as a method for combining fluorescent proteins with histological stains in order to observe cell wall components. Although this paper uses Arabidopsis roots as their study organism to show the histological stains that they will be using, these methods were adapted to woody stems that we are using in this project. For those who may not know what ClearC is, it is a solution that is used to clear plant tissue and reduce chlorophyll autofluorescence, and it does not affect fluorescent proteins, which is great. ClearC is made up of several components, and this protocol listed below can be adapted for as much or as little of the solution you may need. The preliminary stains that we have decided to test are basic fuchsin, which highlights lignin, direct yellow 96, which is a general wall stain and is abundant to xyloglucan, Nile red, which highlights suberin, direct red 23, which is also a general wall stain, but it is instead abundant to cellulose, Oramine O, which highlights lignin, suberin, and cutin. And you are actually able to combine these dyes in order to be able to observe these different cellular anatomy structures. So as you can see here, there's an outline of the combinations that we we'll be using, which are basic fuchsin and direct yellow 96, Nile red and direct yellow 96, direct red 23 and Oramine O. And as you can see from the image on the right, the first is showing direct yellow 96 in yellow, direct red in magenta, basic fuchsin in red, Oramine O in green, and Nile red also in magenta. This image right here is staining of the differentiated Arabidopsis roots that are presented as examples. The way one would approach staining a cross-section is as follows. A pipette with your dye of choice is made in solution with the clear C solution that you will make beforehand, and you will obtain your Castanea cross-sections using the Compresstome vibrating microtome. Then each of the cross-sections will be put into a vessel and stained for an allotted amount of time. Once the cross-sections are stained for their allotted amount of time, you will wash stained cross-sections by gently removing them and placing them in a new vessel with just clear C, no stain, and you will use a shaker or rotator to gently wash sections for this allotted amount of time. The allotted time for both staining and washing the cross-sections will become apparent on the next slide. So depending on what stain you are using, whether it's basic fuchsin, Nile red, Oramine O, direct red 23, or direct yellow 96, there are different staining procedures. So for basic fuchsin and Oramine O, you will have to stain overnight in order to see the results that the dye results, as opposed to Nile red, and direct yellow, which only need a 30-minute span of time to be submerged in the clear C and dye solution. Each of the solutions are then divided into their own specific concentrations, as you can see, between 0.05 and 0.20%. And then the clear C wash time and solution exchange is as follows. When you are staining with a dye such as basic fuchsin, you will wash it for 1 hour, but within that 1 hour span of time, you will change the clear C solution 3 times in order to make sure that your sample is fully clear. And that's the same for the course of the rest of the dyes, except for direct red and direct yellow, which don't need to be washed at all. For double staining, which I will show you an example of in a couple of slides from now, you will want to initially stain sections using a dye that requires an overnight staining first, and then use the second dye and complete the wash after. After staining, cross-sections are then mounted on microscope slides with cover slips to prepare to be analyzed under confocal microscopy. With the support of the plant biology department at the University of Vermont, Dr. Philip Linthalack and PhD candidate Connor Lewis, we obtained preliminary images using a dragonfly spinning disc confocal microscope from Andor Oxford Instruments in conjunction with Fusion desktop software. And these are just some examples of the preliminary images that we have obtained so far. So as you can see on this slide, this is just a single stain where we can see basic fuchsin, direct red, and Oramine O. And again, just to reiterate the points, that basic fuchsin highlights lignin, direct red is highlighting a general wall, but also abundant cellulose, and then Oramine O is highlighting lignin, suberin, and cutin. As you can see in some of these images right here, especially in the basic fuchsin, this is staining the epidermis and the phloem fibers and staining the xylem. Within this direct red image, it looks pretty overstained, and it is staining pretty much the entire cross-section. And in the Oramine O image, you can see the epidermis, and it is definitely staining the suberin as well as lignified phloem fibers. So one thing that we will need to continue to develop over time is trying to figure out what accurate stain concentrations will produce the best images for us moving forward. Here are 2 single stain examples for the remaining stains that we are using, Nile red and direct yellow 96. And again, now red is highlighting suberin, where direct yellow 96 is a general wall stain and is abundant xyloglucan. These are quite exciting images that I really love to share with other people. And these are the confocal microscopy images showing a double stain combination. So the first image is basic fuchsin and direct yellow. The second image is Nile red and direct yellow 96. And the third image is direct red 23 and oramine O. And again, at the bottom of each of the images, you can see a just very quick explanation of which dyes, of which like anatomical features are being highlighted by the dyes that are being shown. So as for next steps for the project, we are continuing to develop our protocol by identifying 2 or 3 more fluorescent dyes to highlight necrotic or fungal cells, callose and reactive oxygen species. We are also going to move forward with inoculating and infecting young American and Chinese chestnut trees in order to survey stems in a time series, as I mentioned before. We will use the combinations of different fluorescent dyes, as I just showed you, to understand the changes in cell wall anatomy and biochemistry over time. Ultimately, we will take our findings and compare it to similar data, specifically investigating gene expression and said biochemical data. And from this, we are hoping to learn and possibly determine if these factors are a basis for resistance versus susceptibility between the American and Chinese chestnut. And with that, I wanted to say a big thank you to the Groover Lab, my PI, Andrew Groover, as well as my lab mates, Nicholas Darcy-Marr and Tegan McAllister, who are in the room with me right now. I would also like to thank our collaborators, the American Chestnut Foundation, specifically Kendra Collins, who works in the same lab building that we do, as well as Dr. Jared Westbrook. And then a final shout out for the endless hours of confocal microscopy help and support from Dr. Linthalak and Connor Lewis from the UVM plant biology department. And with that, thank you so much. And I am open to answering any questions people may have.
That's wonderful. Thank you, Grace. And I love the fluorescent images that you shared.
Thank you.
I want to open up the floor now. I don't see any questions in the chat box, but please feel free to unmute yourselves or show your video. If you're feeling shy, go ahead and enter in the chat box. I'd be happy to ask it for you. Grace, I have a quick question, which is just a more speculative, but given our current struggles with climate change, what do you foresee as the future and impact of climate change on the fungal blight situation for our chestnuts?
That is a great question. And I think it certainly depends on how the fungus functions as a living organism in itself. And so there are some types of microbial fungi that can exist in like certain different like harsh conditions, and those are known as extremophiles. And there are also some that might not be able to withstand those changes in climate change. However, I'm not entirely sure per se how Cryophenectria parasitica is going to be affected over time, but certainly with the abundance of Cryophenectria that we're seeing in our chestnuts over here, I'm not sure if it will be going away anytime soon, but there are certainly a lot of efforts using biocontrol that the American Chestnut Foundation, as well as other federal agencies that are doing in order to repair the damage that this blight has caused over time.
Right. Does anybody else have any questions? If not, I want to thank you again, Grace. And Grace, your actually will be starting her graduate studies in a few months. And I want to say an early congratulations. And we hope to have you back.
Thank you so much. I really appreciate it, Abby. And thank you again to all the participants who joined us this morning. This is my first talk. So I'm very excited that you all attended. And thank you again.
We're doubly excited. It's our first plant talk. And what beautiful images that you shared with us. Thank you.
Thank you so much. Goodbye, everyone.
Bye-bye.